Thank you, Halton, and good morning, everybody. Yes, I'm going to talk about genetic techniques, genetic monitoring techniques for crown-of-thorns sea stars, which is a topic me and a group of collaborators at Ames work on for 7 or 8 years now, and had a project in as part of the integrated pest monitoring NESP. I don't have to go much into details of COTS history. So you all know we're in the 4th crown-of-thorns outbreak since the 1960s, and that crown-of-thorns, when Katharina Fabricius and Glenn Diaz modeled it in 2012, contributed to 42% of the overall coral loss. Despite being in the 4th outbreak, there are still quite a few questions we have with regards of monitoring. For example, we have described the initiation box, as Dave has mentioned before, but we don't actually know where exactly inside that initiation box those outbreaks happen, which is quite important to understand different hypotheses of outbreaks, for example. And we also don't know if the initiation box is actually quite a little bit further north. With regards to stakeholders, it's quite obvious with the crown-of-thorns that the main stakeholders is AMTO and other tourism operations, and Gebrumpa. Of course, Gebrumpa last year published a strategic management framework for crown-of-thorns, splitting the outbreaks in 4 different phases, which is the non-outbreak phase, the initiation phase, the waves of secondary outbreaks, which is the one we are currently in, and then the termination phase. And within that document and in other discussions, it was clearly defined that specifically during phase 2 and also 3, early detection and monitoring is extremely important, and that early detection, only early detection of the outbreaks can warrant early intervention. So what is the problem we're trying to tackle? Well, the main problem is that standard current monitoring techniques have quite a few flaws and problems and won't give us the right results at those low levels we're looking at. Firstly, normal scuba-based surveys are not too bad, but they cover quite a small area. If you are looking for COTS more than 25 centimeters, you have a probability of 80% of finding them. However, if you go below that, Aaron McNeil et al. 2016 modeled, you pretty quickly drop your likelihood of finding the COTS. Secondly, Morgan Pratchett has shown us some preliminary data at the COTS conference 4 weeks ago here, that at low densities, below 5 per hectare, the likelihood of actually detecting COTS, even if you know they're there, are extremely low, are below 50%. And this is still in scuba surveys. If you're going to any toe-based technique like the manta toes, citabilities are probably less than 20%. And remember, that's 20% of what you already see, that it's 20% of maybe just 50%, which goes below 10%. So that's quite bad, so there's an obvious need to improve on that. One other problem is toe-based techniques like the manta toe usually toe the reef perimeter. This is Davies Reef, just as an example. We have examples of a few more reefs. Davies Reef is a reef which is part of the LTMP team, which is manta toed frequently every second year probably. It's got 40 to 50 toes going around the reef. But those toes are just on the reef perimeter, remember that, because those areas are easily accessed. All other reef areas are quite hard to access using a Zodiac or any other vessel and tow something behind you. So if you look at the manta toe data from that reef, basically over the last few years there was just a couple of COTS detected, and the toe densities are less than 0.1 per toe, and 0.1 is defined by Gebrumpa and in the management scene as a no outbreak area. So these are basically no outbreak reefs, supposedly, because you can't find anything on the manta toes. However, if you're looking closely, we did fine scale surveys on the reef. you find really big aggregations on areas which just can't be towed. So we're finding areas here near the weather station, for example, in the thickets of the corals where we have 15 to 20 cots per hectare, so far above those threshold levels David mentioned before. And we've got those data for a few reefs now. And the biggest problem, well, one big problem with underwater surveys is occupational health and safety. As you can imagine, manta towing has been likened to fishing with a lure. Basically, you have a pretty big lure up there, and you've got big predators out there. Another big problem is not just sharks, but crocodiles, because, and possibly a reason why we haven't looked in much more details further north, like Princess Charlotte Bay, is you have significant crocodile hazards towing people over the reef or even diving down there. In actual fact, I just talked to Kate Osborne from the monitoring team because we're keen to survey Princess Charlotte Bay with our techniques in a couple of weeks. Two reefs which are known outbreak reefs are basically taken off the reefs of the list of the LTMP currently because they can't be surveyed because of crocodile risks. So that's another slight problem. So our task and our was to do field assess eDNA-based technology to improve monitoring and early detection of COTS using eDNA. Just briefly, eDNA is called environmental DNA. It could be free, dissolved in the water column, or absorbed to surfaces like sediments, or still in cells and floating in the water. It's been used quite a lot in terrestrial and aquatic environments over the last 10 years, for example, for biodiversity descriptions and detection of introduced species, or food item identification, such as those techniques Federica has just talked about in fish guts. And it's more recently also been used in caves to find Neanderthal DNA and describe population dynamics of Neanderthal populations, which is a bit off topic. Anyhow, so we have basically developed the methods to specifically detect COTS quite like 6 years ago. When we talk about COTS DNA, we have two different main applications which always confuse ourselves and listeners to our presentations. We have what we call the plankton DNA, so that's looking for larvae in the plankton. That's when we're going out in summer season, during the reproductive season. We take plankton samples with plankton nets, and we then quantify the amount of larvae in those. And more recently, we have moved the technique also to test for adults or post-settlement adults and juveniles on the reef, which we do in very small 2-liter water samples, but we've got to collect quite a few of those, which are filtered over a filter and then extracted, and DNA is identified with PCR-based techniques on those. Additional applications are what Federica has described. So Federica has already used this for food predator detection, and Morgan Pratchett and I have a student, Peter Doll, who's working on settlement traps to quantify settlement of COTS on the reefs. We're stuck. So the first few results I'm presenting are on the plankton part, or the left-hand part of that. We do, since we did our first surveys in 2014, so we do an annual AIM survey. However, that would also represent 10% of these samples. What we see here is not positives or negatives. Every blue dot is simply a sampling location we have sampled with the plankton samples, which is ready to be analyzed, or some are analyzed. We have an amazing data set because we have an amazing amount of helpers and collaborators. Basically, 2 pontoon operators are helping us to collect plankton samples. All COTS boats are collecting plankton samples for us. RRC and MTO collects, and also the Joint Field Management Program collects. So we have a large data set. We're contributing to it ourselves, and we have the ability to quiz these data for all sorts of questions, mainly for where the larvae are and how they're distributed. So again, this is just plotting them on a north to south gradient. So this is latitude here versus time. If we're zooming in now into just the year before last, so the year before COVID, we analyzed those data. And so this is just the spawning season 2019-20. What you can see here is now blue dots are negative results, whereas green dots are actually positive results for larvae in the plankton. What's quite interesting is that we find a lot, and the larger the dot is, the more larvae we find. So we're finding a lot in the Townsville region, which is really no surprise, because that's, as we had heard before, that's where the current COTS outbreak basically is, and so that's nicely reflected in the larvae. We also find a few COTS larvae in the Cap Bunker group for the first time, which is good, and there are a few outbreaks reported, not in high numbers, and they're culled down there. But what's really surprising and really encouraging for us is that we're actually finding COTS larvae in the plankton at the Lizard Island area, and that's an area where there's hardly any COTS found in the moment. So that means it's a very, very early detection for COTS we have here. What's plotted here is the regional manta toe averages. So again, we're above threshold level here in Townsville, so no surprise to find them here, but below levels here and at extremely low levels here at Lizard Island. So yes, I think plankton eDNA can be used for regional, very early detection at very low densities, and then guide us to the right regions to focus in with control. Now, coming to the juvenile and adult samples, so with those we're taking many samples. Currently we're taking all samples ourselves. We have two reefs which we followed over the last 3 years, which are Davies and Bowden Reef, which we have chosen because of the reasons I've shown before. Basically, they would be classified as non-outbreak reefs because they have very low manta toe data, but they have a few COTS sitting on the reef. So you see here, this is the number of samples we're taking per year on the reef. So it's between 50 and 60 on Davies Reef, and it's between, well, the same number on Bowden Reef. So that's quite a lot of effort at the moment. But what we find on these reefs is that this is the percent of positive samples. So 80 to 90% are positive on Davies Reef and slightly lower, but increasing numbers on Bowden Reef. We can take those data and do a little bit of statistical modeling with it, which is called occupancy analysis. So you can basically, here's the sample size is on the x-axis and the probability of detection on the y. So this models, if you have only one sample taken, how big is your probability of finding a COTS? And then you can multiply it out to what would happen if I take more than one sample. So what's really quite surprising to see, if you're looking to detect COTS on reefs like these two non-outbreak reefs, you would actually only need 2 individual water samples to find that there's positive COTS above detection levels. Even more surprising, again, over the last few years, we've put a lot of effort in getting up to Lizard Island because of those reasons, that it's got really low COTS densities. And Morgan Pratchett surveys using scooters over the last couple of years showed that there are clearly COTS there, but they're below the threshold levels Dave mentioned before, the reproductive threshold of 3 per hectare. So we've nearly taken 200 samples every year there. We see many less are positive than the ones on Bowden Reef and Davies Reef. And however, if we do the same type of modeling, we still show, even on Lizard Island, only 10 water samples of 2 liters would be sufficient to detect COTS at those low densities. So that's minimum effort, which would really be needed. However, that's just a detection. There's a few different methods to apply these methods, of course. This would simply be a detection asking whether or not they're above those threshold levels. We would probably be interested in an actual quantification, which would require taking a few more samples and taking into consideration the effort you take going to the reefs. It would probably be silly just collecting 10 samples and go away again after 10 minutes. However, these are strategies which can be fine-tuned and finalized. Our current analysis is in progress, so we will likely recommend something of in the area of collecting 3 sites per reef and taking 15 samples, which is still an exercise which can be done in a couple of hours. So even with this higher effort, we could survey 2 to 3 reefs a day. If we would go to this lower effort, we could practically, well, the only limitation would be the speed of the boat we've got. So we could do 6 reefs per day probably with this way to service it, to survey it. This is a little bit slow. Anyhow, I hope I gave you a quick overview and impression of the work we've done. Our conclusions are that we think larval DNA is a powerful tool for early detection on a regional scale, so no way this is for the reef scale. However, it still provides really good guidance of where the larvae are, where they're likely to settle, and where we should focus our effort to actually monitor it. There are a few interesting things, like we don't know why those larvae are in Lizard Island. Again, that's completely open to interpretation. Some people might say that could actually indicate the start of the next outbreak. Maybe it's just remnants of this outbreak. So only surveys over the next few years will show us what this means. Additional benefits of, then we have this large network of collaborators who are collecting with us and who are working together with us, which is really big help and which makes it quite cost effective to conduct this monitoring and research. Additional benefits of the larvae work are that we also learn more about the ecology of the larvae, where they're sitting in the water column, how the connectivity is between reefs, which would ultimately help us to further predict outbreaks and the spread and connectivity between reefs. The post-settlement or adult eDNA, we think, is an ideal warning tool for surveys on the reef scale. We are currently, with the help of Barbara Robson, also investigating sub-reef scale detection, whether that's possible or not. But that's something we will look at over the next 6 months probably. So we're pretty sure we can detect COTS with high likelihood below those thresholds which were mentioned before. eDNA monitoring is a good alternative, I think, and it circumvents those 3 problems: detectability, the untowable areas, and occupational health and safety. In the longer term, or not so long term, it will also be possible to train collaborators, people from the industry or citizen scientists to take those samples for us. We have developed with quite easy techniques which are called self-preserving filters. So you literally just have to filter your DNA, put it in a bag, and you could send it for analysis. So that's quite easy. In summary, we believe that both adults and larvae detection are important new monitoring tool, and again, this mantra that we think early detection in the future will allow early intervention, which might prevent future outbreaks. Thank you.